Hi, my name is Nadav. I'm from the Hebrew University of Jerusalem, and today I want to tell you about our recent work, quantifying gene selection in cancer through protein functional alteration bias. So in this work, we developed an algorithm to find cancer genes. When I say cancer genes, I mean any gene that is involved in some way in cancer. So it could be, for example, oncogenes or tumor suppressors. And the way to find those genes is to detect signs of positive selection in tumor. So within tumor, we expect those genes to accumulate genetic alterations. Now, the classic way to find those mutations is what we call the frequentist approach, which is basically just a counting game. So here we try to look for genes with excessive mutation rates. So this means genes that have more mutations than you would expect under normal conditions. Now, in order for this to work, you need to define what it means to have normal mutation rates. And we know that actually mutation rates vary quite substantially across different samples and cancer types and genomic loci, meaning that in order for this method to work, we actually need to model all of these variables and how they affect mutation rates. And this leads to actually many assumptions of the frequentist methods. And when we finally do find a suspect gene, it's very hard to tell whether it's a genuine cancer gene or maybe just some of the assumptions are inaccurate. So we propose what we call the functionalist approach. And here the idea is instead of looking at the number of mutations, we will actually look at their content and specifically their functional effects on genes. And we will try to highlight those genes with mutations that appear to be more damaging than expected at random. So we developed Fabric, a new method that works in two simple steps. So first, given the list of observed mutations, it will try to assess their impact on protein coding genes. And then second, it will try to find those genes that seem to be more damaged than expected. And I will say that this work was actually recently published in Nucleic Acid Research. So if you want to read and learn more about the method, you can find it there. And actually it has the exact same title as the title of this talk. Okay, so the first step, we want to assess mutation impact. So the way this works, we take the list of variants and we filter those variants within the coding sequence of the gene, and we see what each variant seems to be doing to the sequence of the protein. And then we assign it what we call an effect score, so a number between 0 and 1, which tries to estimate the functional effect of this mutation and whether it's damaging or not. The way we assign those effect scores is through a machine learning model that was pre-trained on the CleanVar dataset. And importantly, this dataset is completely independent of the cancer cohort that we then used with Fabric. So for each variant, we extract different functional and biochemical features, so over 1,000 numeric features overall. And we then feed them to a standard machine learning algorithm and get the predicted effect score for the variants. Okay, so the second step is to find genes that seem to be more damaged than expected. And for that, we need a background for the effect scores. So here we can actually see a real example from the P53 gene, maybe the most famous cancer gene. Okay, so first what we do, we look at the observed mutations and calculate the frequencies of all the 12 possible single nucleotide substitutions. And then for each one of the 12 substitutions, we calculate its own background distribution. So for example, for the A to G substitution, we will simply go through the coding part of the gene, through all the A nucleotides and substitute them with G, and then feed it to the exact same underlying machine learning model and record the obtained effect score to get those distributions. And then we mix those substitution frequencies with the distributions of the substitutions, and we get the overall background distribution of effect scores. So this describes for a random mutation within the coding region of the gene, according to these substitution frequencies, what would be its expected effect score. And then we can compare the effect scores of mutations that we actually observe, what you see in orange, to this background, what you see in blue. And as expected, for such a dominant cancer gene, the observed effect scores are much more damaging than expected at random. And we can actually quantify it. So Fabric can assign a z-value to measure the strength of the association and a p-value for significance. So what are the strengths of this framework? So first, it's designed to really make minimal assumptions. So unlike the frequentist approach, which needs to account for all kinds of variables, which might influence mutation rates, in our case, the number of mutations is already given, so we don't need to account for all of these variables. And this really leads to minimization of false discoveries. And importantly, even under inaccuracies of the underlying machine learning model, which is of course inevitable, no machine learning model is ever perfect, since we use the same model for both the background distribution and for the observed mutations, we know that those occasional errors will not lead to systematic biases. So it can diminish the statistical power, but it shouldn't lead to false discoveries. Now second, it's fully complementary to frequentist methods. Okay, so one class of methods look at the number of mutations. We look at their content. So under the null hypothesis, we know that the p-values that they get should be completely independent because they look at completely orthogonal biological signal. And that means we can actually take the lists of genes that we get from them and compare them, which would be very powerful. And we can also do a meta-analysis. And finally, again, because the number of mutations is not a factor in this approach, we can also find genes, cancer genes with low recurrence. So we used Fabric and analyzed about 3 million somatic mutations from approximately 10,000 individuals from the TCGA cohort, covering 33 different cancer types. And we found almost 600 significant genes showing positive selections. And actually, many of them are new discoveries of genes that hadn't before been associated with cancer. And even though the method is completely symmetric, so it can find negative selection just as much as it can find positive selection, it only finds six genes with negative selection when we give it somatic mutations from cancer, which is what we expect from tumor. And it can also be used to highlight differences between cancer types. So here you can see the z-value that Fabric assigns to different genes across different cancer types. And we can also take the differences between those z-values. So here for each gene, you can see those cancer types that have a z-value which is significantly different than all other cancer types. So this is a very neat way to highlight cancer type-specific patterns. These days, we also developed the Fabric Cancer Portal to make those cancer results much more accessible. So you can look for any gene of interest out of the 20,000 protein-coding human genes. And you can get all the summary statistics of Fabric across all the different cancer types. And you can compare different cancer types and different genes. It's also worth saying that you can use Fabric in other evolutionary contexts as well. So for example, if instead of cancer you feed it with germline variants from the human population, you will actually observe the exact opposite trend. So here in red you can see cancer genes, which are mostly dominated by positive selection, as opposed to standard variants from the human population, which are dominated by negative selection. And as expected, this also correlates with allele frequency. So for example, for common variants, we see that they tend more to negative selection. And it also works in other options. So for example, you can use it to analyze de novo or rare variants from some cohort, say with a proband with some condition, and to detect genes, again, that appear to be more damaged than you would expect. And more generally, Fabric can operate on any set of single nucleotide variants, and it will give you genes that appear to be more or less damaged than expected. So it's actually very easy to use, and Fabric is completely open source. So here on the bottom, you can see the link to the GitHub page of Fabric. and it has a very simple command line interface that you can use on any data set. So I will conclude with that, and I want to thank my two advisors, Michal Linial and Nadav Brandes, and I also want to thank Or Tsuk and Guy Kellman for their help. And thank you very much for listening, and I would be more than happy to take questions.